Second question is where they live, right? So we will deal now where do they live, right? In a way, you can think of the marine zonation. In the last week, we have seen that this is the marine zonation. You have the neritic zone, you have the oceanic zone, in you have epipelagic or photic zone. Then you have euphotic zone, right? You have the continental shelf, right? I hope that you remember these things. You have the sea floor, you have the intertidal area, I mean, sorry, continental shelf, and then you have the intertidal area, right? So in all the habitats, microbes are present. Different microbes have occupied the different areas. Microbes can, I mean, some of the species can be ubiquitous, that is, it is present in almost all the surfaces. Some of them have occupied the photic zone, for example, phytoplankton, because they require, or any autotrophic prokaryote, because they require I mean, sunlight for the photosynthesis. Some of them are there in this particular mesopelagic region doing the what? Hope you remember last week I talked about the this microbial loop, or the utilization of the dissolved organic matter in the water column, right? So you have that particular class of bacteria or microbes here. Then you have in the continental slope and the continental shelf different set of microbes performing the activities or the nutrient cycling in the sediments, right? Then you have a whole set of bacteria or microbes specialized in the hydrothermal vents, and of course in the intertidal area, right? So you can see that microbes are present in all the environment in different areas, right? In a nutshell, we can say that phytoplankton in the photic zone, or you can say that any bacteria which is depending on the sunlight for the photosynthesis will be there in the photic zone, and photic zone means anything in this area, right? Then you have the associated with the suspended particles, colloidal materials attached to the surfaces like rocks, submerged structures, right? In all the places. Also, it is having the association with the plants and animals that we will see in the next slide, and then in the sediments, right? So this is a very nice picture to understand that if you look through the microscope, maybe not with the magnifying glass, but with the microscope, any even 1 ml of the water from the surface, you can see that a lot of photosynthetic bacteria or photosynthetic microbes are there, which utilizing the carbon dioxide and the sunlight produces the oxygen, right? So this is a representation of the bacteria present in the, or microbes present in the photic zone. Then we have in the water column, as I was saying, in this particular area where the bacteria are associated with the dissolved organic matters, right? And these bacteria, I mean, feeding on or utilizing that dissolved organic matter, either making it into the recalcitrant dissolved organic carbon, or otherwise it is going back into the microbial pool, right, or the viral cell. Hope you remember this, right? So so we can say that even in a single drop of seawater, there could be close to 1,000 small protozoans, there could be 10 million viruses, and there could be at least 1 million bacteria. So all the seawater is full of microbial life. So this is about the surface, this is about the water column, okay? Now in the sediment, we know that they perform a number of functions which is exclusively done by the bacteria. For example, in the carbon cycle, in the sulfur cycle, in the nitrogen cycle, and so on, right? And this is another slide, and the repetitive slide, where it indicates, or where it further emphasizes that even in this particular subsurface or subdeep surface, you have a plenty of fraction of biomass of the bacteria or the microbes, okay? So it means it further establishes that the microbes are available in all the habitats in the ocean. uh, Not only in the normal water, in the normal water column and the sediment, also it is present in the extreme environments. So extreme environments, you can see that there are hydrothermal vents, there are shallow underwater hydrothermal vents, there are deep sea hydrothermal vents, right? In all those locations we do have bacteria. And also one particular example I gave last week for the oxygen minimum zone. So this oxygen minimum zone also have set of nitrogen cycle bacteria, which has been performing the fixation of the nitrogen in the oxygen minimum area, right? Then you have shallow water hydrothermal vents, or you can say that is a hot springs. So this is one example of Forio, which is in Ischia Island in Italy, where the whole island is. Last week I showed you how the volcanic island looks. So that was Ischia, and this is a part of Ischia where you can see that hot water springs are there. Hot springs are there in the seawater. What happens due to the volcanic activities? There are vents uh, from where the heated water is coming into the surface, and people are actually taking the therapies or sauna, open sauna in the sea. Okay, so here also we do have a rich microbial biodiversity. Another example in Paneria, again in Italy, you have several locations where you can see that the bubbles are coming out in the water, and it brings not only the sulfur or not only the toxic metals, but also the hot water. Okay, so all these locations are hydrothermal vents, shallow hydrothermal vents, making the hot springs. Okay, then again, I will give you some examples that microbes are also present in the hypersaline environment or in the salt pans, where the salinity goes at least 10 times higher than the seawater. That in normal seawater we have 35 ppt, that is parts per thousand, and in the hypersaline area, it will cross close to 300 or 400. So you can understand 300 ppt, that is 10 times more than the marine environment. In a nutshell, you can see the microbes are present in the sea ice, polar region, in the ice sheets, in the volcanic seeps, in the hot springs. I'm not going into the terrestrial part, uh, but I will just focus here on deep sea hydrothermal vents, uh, in the sediments, deep sea sediments, in the continental subsurfaces, in the hypersaline lakes. So in all the locations, we do have a thick varieties of microorganisms living. Okay. Uh, Another example that we know that bacteria are present not only in the human gut, or only the terrestrial animals, but it is also abundantly present in the guts of the marine fishes, and for that matter, for all the marine organisms, right? So there is a very recent paper about this in a Frontiers journal in 2017, no, 2018. Similarly, the sponges, which you may be remembering from the class of Prakash or from the class of Dinesh, that sponges are there which is covering to the corals, right? So those sponges are totally associated with the bacteria, and in some cases they are 80% bacteria than the sponges, right? Also, there are corals, which are, there are bacteria which are associated with the corals, performing several functions and kind of a symbiotic relationship between with the corals, right? Last week I gave you example of the deep sea adaptation, that there are ESCA, right, in the anglerfishes. So in the anglerfishes, the ESCA is light, is having the light, right? So this is nothing but the bioluminescent bacterial colony which performing the quorum sensing and this operon for the luminescence, okay, and producing the light. So this is a very good example that bioluminescent bacteria associated with the fishes, and we know that how efficient this, or how important this light is to run the food with, right? And then also we have the seaweed associated bacteria. So you can see that this is one leaf of the seaweed done the microscopy, and you can see that so much of bacteria, viruses, and other microbes are present on the surface of the leaves. And these microbes plays an important role, because if they are habitat of that particular leaf, it saves the seaweeds from the infection of the pathogenic bacteria in the marine environment, right? So if the host microbiota replaces somehow due to the temperature or due to some biotic or abiotic interactions, then pathogen may invade this area and can get over the seaweeds, causing the disease in them, right? So this is how all the marine organisms, marine microbes are present in not only in the different habitats of the ocean, but also interacting with all the living organisms in the marine environment, right? Now, stopping here, I will tell you two important concepts. One is sea surface microlayer. So, sea surface microlayer are, if you see in this particular image, the surface where the seawater is interacting, or the layer where the seawater is mixing with the atmosphere, there forms some sea surface microlayer. So this sea surface microlayer can be 1 millimeter thick, right? And it is because the physiological and physiochemical and the microbial uh, aggregation from the phytoplanktons. So so the phytoplanktons, right, produces the polysaccharides that is also called as transparent exopolymer particles. So these transparent exopolymer particles will go and resides on the surface of the water where the interaction with the atmosphere is happening, and causes a close to 1 mm thick layer. Okay, so that is called a sea surface microlayer, and this you can understand it as a kind of a microhabitat, because there a very distinct class of distinct varieties of microorganism live in that particular microlayer, because it is rich with the exopolymer particle, which is rich in the polysaccharides. Okay, that you can also think as a gelatinous biofilm on the surface of the sea, right? Now, last week I gave you one question, that what is newstone, right? And then some of you have answered that newstones, right, are those organisms which are present on the surface of water, but it does not go in, right? And then I said that due to the this surface tension of in the water, that water is strider, one example I gave you, right? It is because of this gelatinous biofilm layer of sea surface microlayer, those organisms can easily attach to the surface without going in, or without keeping there, or without interacting uh, with the water column, right? Now I hope that it's clear that these microbes produces the polysaccharides, which can be also seen as a transparent exopolymer particle, and then it comes on the surface, forming a sea surface microlayer, which is a kind of micro environment for a distinct set of microbes, right? And it is gelatinous in nature, and because of this gelatinous biofilm, that phytonewstone or bacterionewstones are staying on the surface of the water, right? There is another important concept, that is a biofilms and the biofoulings. Many of you are aware that when there is a ship, or when there is any sessile structure put by the humans, or even in the nature, or for example in the rocks, then what will happen? A number of organisms will come and attach to it, forming a biofilm, right? Correct? So what happens? What is the procedure? Initially, let us suppose we have a ship here. In the ship, what will happen? A certain set of bacteria will start coming, and then it produces the polysaccharides, or it produces the extracellular matrix, right, which is rich in the polysaccharides, DNA, and other biomolecule content, right, which attracts other bacteria to come and settle, thereby making a kind of a layer of the exo polysaccharides, right, or extra uh, exo macromolecular, you can say. Then more and more bacteria will come. Now this will make a kind of what do you say, a biofilm for the bacteria. Now, utilizing this extracellular material, barnacle will come there and start settling on the gelatinous substance, right? Similarly, algal spores will come and start settling. As a result of which, what will happen? You can see in this particular image that the primary colonization is by the bacteria, which has helped the secondary coloners in the I mean seaweeds and the bivalves and other stuffs to come and settle on the surface. This is a very important concept because biofilm leads to the biofouling, and biofouling is a major area of research in the marine environment because biofouling causes a lot of wastage of fuel in running the ships because that increases the weight of the ships, and as a result there are several paints. Frequently you have to paint them, frequently you have to change the coating of the surface, and so on, right, in order to avoid the biofilm and the biofouling. So again, as I said, that this biofilm is being created by the marine bacteria or the marine organisms, right? So now I hope that two new concepts you have got today. I hope first is the sea surface microlayer that is being made by the extra exopolymer particles of by the phytoplanktons on the surface of the sea. And the second one is the biofilm and the biofouling. That is, the bacteria will get attached to the hard surface. It secretes the exopolymers in the form of exopolysaccharides and DNA and other component. It attracts the other bacteria through the quorum sensing, and then once there is a thick layer of exo secretions, then barnacles and algal spores and other things will come and settle on that, and then it causes the biofouling, right? So you can see a paper by Martin Rodriguez, which has nicely explained the particle of organic matter, then primary colonizer, secondary colonizer, tertiary colonizer, and then you have the microscopic fouling communities. Okay. So what you understand with this particular slide? So where were we? We were there that where they are living. So as I said that they are living everywhere. They are living not only in the mesophilic conditions or the normal conditions, but also in the hydrothermal vents, in the hot springs, in the cold waters, in the salty waters, in the high nutrient rich environment, right, or high heavy metal environments, right, and not only on the environments but also inside and outside the organisms. Some examples are the gut microbiota, bioluminescent bacteria on the Angela Risca. Then you have the sponge associated, coral associated, and then seaweed associated, right? Then I give you two concepts that bacteria not only are present everywhere, but it also changes the microhabitats. For example, the phytoplankton releasing the exopolymers, forming the sea surface microlayer, right? And then this sea surface microlayer will have a completely distinct variety of the microbes. Second is the development of the biofilm. So it means that they do have a communications and communication through quorum sensing, forming the biofilm, leading to the biofilm, right? A question remains that though they are present in all the life, I mean, in all the habitats, in all the life forms, everywhere in the marine environment, what do you think? If the microbes are evenly distributed in world ocean, or can you say that if the microbes are present, the same microbes are present in Indian waters and same microbes are present in Caribbean waters, or what do you think that regulates the diversity of whether if first question is that whether there I mean the microbes are evenly distributed, second if it is not evenly distributed what could be the reason, and third that whether there is a temporal variations of the same species in the different part of the ocean. Anyone can answer this question maybe in a brief? Yeah, so a whole lot of physical and the chemical factors and also biological factors which regulates the distribution of the microbes in the world ocean. Of course, by saying this, I am not saying that if one species you are getting in India, that will not be present in, let's say, Spanish water or somewhere, right? But what I mean is, what I mean by saying this is that in some certain area, some community structure will be dominant, and in some other areas some other community structure will be dominant. And one particular example is that let us suppose that considering any abiotic factors, right? So if the bacteria is more adapted to live in the cold waters, that may be rare chances that that will be available in the other part. But again, the question is that most of the current systems and most of the waves, and we have seen a lot of physical activities in the sea that can bring the microbes from one part to another. And again, we have to keep in mind that there are microbes which are adapted to a certain conditions, right?